Wenyi Feng is associate professor in the Department of Biochemistry and Molecular Biology at SUNY Upstate Medical University. She received a PhD in Biochemistry and Molecular Biology in the University of Miami, Miller School of Medicine in 2002, studying DNA polymerase epsilon in fission yeast with Dr. Gennaro D'Urso. Wenyi then commenced a postdoctoral training with Dr. Bonnie Brewer in genome science at the University of Washington. And since joining the faculty at Upstate in 2010, Wenyi Lab has been studying genome instability, particularly in the form of DNA double-strand breaks induced by replication stress in multiple experimental models. Finally, I want to stress that her lab invented break-seq technique and has made key seminal contributions to understanding a novel genome maintenance function of FMRP, the protein responsible for Fragile X syndrome. So without further ado, please, Wenyi, whenever you're ready.
Thank you so much, Inés, for the lovely introduction. It's my pleasure to speak in today's webinar and tell you a little bit about our recent work, starting with the Fragile X syndrome disease interest, and which how it led us to the path of technology development. So Fragile X syndrome is a neurodevelopmental disease that is also the most common form of inherited intellectual disability. 99% of the patients actually has a mutation on the X chromosome that is due to the repeat expansion of trinucleotide CGG, which when it exceeds 200 copies will give the full disease spectrum. And so this CGG repeat is located in the 5' UTR of a gene called FMR1. And when it expands, it will lead to the epigenetic silencing of the gene and complete absence of the protein product called FMRP. FMRP is RNA-binding protein. And as I said, 99% of the patients have a mutation in the repeat expansion, but there are rare cases where missense mutations are reported. And this underscores the fact that the loss of function of FMRP is really the beneath the disease etiology. And prior to our entrance to this field, FMRP is primarily known as a translation repressor. So it plays really important functions at the synapse, where it controls a process called mGlu-R, long-term depression pathway. And in the absence of FMRP, where protein translation goes out of whack, this leads to exaggerated long-term depression. And so 95% approximately of the FMRP is actually in the cytoplasm interacting with RNA, controlling translation. But it does shuttle into in and out of the nucleus, where approximately 5% of the protein is found. And so we kind of stumbled onto some discoveries where we saw that FMRP can localize to the nucleus under replication stress conditions. And we subsequently discovered that FMRP is actually a novel genome maintenance protein, and it controls the accumulation of R loops, which could be harmful to genome stability. And it does so by regulating a group of enzymes that are R loop resolvases. And so with that, it serves as an intro to how we made the discovery in the first place. So after we had performed some break-seq experiments, which were intended as a control, where we assayed the Fragile X patient-derived cells, we made the discovery that these patient-derived cells apparently accumulate the double-strand breaks genome-wide, particularly under the treatment of a drug called fiticoline, which is a DNA polymerase inhibitor. So in replication stress conditions, we have genome-wide accumulation of double-strand breaks, and they're enriched in what are shown here, these neurodevelopmental genes. So that's essentially what got us on the path. And we did some computational prediction and saw that there is a great correlation between these double-strand breaks and R-loop forming sequences. And now, since the publication of those initial papers, we had made now missense mutations that also gives you the same phenotypes that we had recapitulated. So these two mutants will skip the actual nature of these mutations, and here's a wild-type control, where we just assay for R-loop formation in these cells using an antibody called S9.6 to recognize DNA and RNA hybrid. We saw that the mutation, so both FMRP mutants not only accumulate the nuclear R-loops compared to control, but also accumulate cytoplasmic R-loop over control. Now, some of you probably are aware of the fact that S9.6 antibody can be non-specific and it detects double-strand RNA as well. And so we are aware of that, but the reduction of the signal by RNase 3, which removes these double-strand RNA, doesn't actually remove all of the R-loops, the signals in both compartments. So, and then the last part I want to tell you about why we're interested in detecting R-loops, particularly in the cytoplasm, is that through transcriptome studies comparing Fragile X patient cells to a control, we saw an upregulated pathway that suggests the innate immune response pathway is upregulated in these patients, which is indicative of the accumulation of cytosolic nucleic acids. And we wonder if R-loops in the cytoplasm is actually triggering this response. So we wanted to now, ultimately, we want to be able to detect R-loops in both compartments. To do so, R-loop detection has been done quite extensively already. On the left, there are these collections of sequencing technology using the S9.6 antibody to pull down the hybrid. On the right are similarly pulling down of the hybrid using the RNA or the hybrid binding domain of the RNase H enzyme, which normally targets the RNA DNA hybrid. And on top, you've got also technology to pull down the single-stranded DNA that's displaced from the DNA from the template DNA. And then finally, on the bottom is a most newly discovered or developed technique that took a nihilist approach to destroy everything except for the DNA RNA hybrid and identifying it at high resolution. So we thought we want another one because the single-stranded DNA seems to be an area that's not as developed. And also, thankfully, we were funded by NHGRI to develop a method to map single-stranded DNA genome-wide. So we thought we could combine these approaches to identify R-loops by a method that we simply call labeling single-stranded DNA short LAS. And this relies on the fact that you can random prime a piece of single-stranded DNA and label it with biotin DNTPs and subsequently pull down those labeled DNA without having to remove any other templates. And we first tested this technique in situ. So we took cells containing these R loops and washing these primer, polymerase, and substrate, or DNA, the biotinylate DNTPs, and then using the anti-biotin to detect its presence. And then in the mock control, we removed the DNTPs. And so, as you can see, the endogenous biotin labels the cells primarily in the cytoplasm, probably around the ER. And with the labeling, now the signal rushes into the nucleus. And then when you treat it with RNase H, it goes away. And we can now also do this genome-wide, which we call GLAS or GLAS, where we embed cells in agros, and then we treat them to review these genomic template, either gaps of single-stranded DNA or R loops. And then we flush it out and capture the single-stranded after denaturation, and go through a round of optional second-strand synthesis and ultimate Illumina sequencing. And here's the genomic view of the signals that we captured, and they form discrete peaks, but also cluster at certain places. And so this part of the work is very much ongoing, but a lot of the candidate sites that we have looked at appear to be long non-coding RNAs, and one of which has been shown to regulate a sort of a parallel protein in ALS, which is TDP43, so parallel to sort of FMRP function in these neurological disorders. So this part of the work is very much ongoing, as I said, and I will end my talk here and thank all the trainees that have contributed to this work, and a shout out to the NHGRI for funding this research, and thank you for your attention.
Wenyi, I have many questions, but one in particular is, why do you think that being FMRP that you discovered that is also part of the double-strand break repair in the nucleus, why do you think there are the particular genes that you're looking at, or that the particular genes that are affected by FMRP mutation are more affected than others? Do you think it's just R loop related? Do you think there is something else? And then, kind of related to that, it has been shown that neural activity actually drives double-strand break, and the transcription factors bring all this amazing machinery to solve the double-strand break and to protect the neurons from this stress. Do you find any relationship in your data to that?
So the breakage that we found in genes, we believe it is driven by R loop. And because it's induced by replication stress, where these R loop accumulation is really, most heightened. And so the enrichment in these neurodevelopmental gene was kind of surprising, because we were actually just using lymphoblasts, not even neurons, which we are doing at the moment. So I can't tell you about, synaptic activities, how it correlates with FMRP breakage sites in our model yet. But as you said, double-strand breaks is actually used to control neuronal activity. So I think the linkage that I know of between our system and that model is topoisomerase, which we can actually co-IP with FMRP, and we think it's involved in R loop resolution.